Upstate Medical University in Syracuse, New York invites you to be the informed patient with the podcast that features experts from Central New York's only academic medical center. I'm your host, Amber Smith. A researcher from the National Cancer Institute recently joined the Upstate faculty, and I'll be talking with him about some of his work. Dr. John Brognard is an assistant professor of surgery and pharmacology at Upstate, and he researches cancer. Welcome to the Informed Patient, Dr. Brognard.
It's nice to be here.
You have a chemistry degree and a master's in biotechnology, but it sounds like you've been focused on cancer since you were working on your doctorate. Is that right?
Yes, and even before that, I did an internship at the National Cancer Institute while I was getting my master's in biotechnology at Johns Hopkins University. And this was in Phil Dennis's lab, and I was there for 3 years, and it was part of my master's thesis. But basically, what we were trying to define were signaling pathways or pathways that really cause lung cancer, and then therapeutic strategies to interrupt those pathways so it would benefit the cancer patients. And one of these enzymes we'll talk about later on is referred to as AKT, but I started studying that essentially soon after I graduated from undergraduate university.
So you were interested in cancer at the same time or before you even knew that you wanted a career in science, it sounds like.
Yes, and I think two factors played a major role in that. I had close proximity to the National Cancer Institute located in Frederick, Maryland. There's a branch there at Fort Detrick. And then also I witnessed my grandfather passing away from lung cancer, and it was pretty devastating. And to see the treatment really, in my opinion, cause his kind of demise and quick deterioration of his health was concerning. And at that early age, I was really impacted by that and thought we needed much better therapies for patients with cancer. In particular, in this case, it was lung adenocarcinoma.
What can you tell us about the discovery you made while working on your doctorate at the University of California, San Diego?
This is a really exciting time, I think, as a scientist, where you're really developing the skill set to really carry forth your career. And we worked on a novel enzyme referred to as a phosphatase. It was the FLIP2 phosphatase. It had never been studied before in humans, so it was completely new and novel. And we discovered that this phosphatase was really critical in regulating pathways that are essential to promoting cancer and the development of tumors. So it was these two pathways and enzymes downstream that are referred to as AKT and PKC, and these tend to be master regulators of cancer growth and tumor growth in general.
And you used the phrase flip in relation to phosphatase. What does flip mean?
FLIP is actually spelled PHLPP, and it's FLIP2. So there's a FLIP1 that was also discovered from that lab while I was there. And it's usually an acronym. The PH part of it stands for a pH domain. This is required for localizing the enzyme in cells. The L stands for leucine-rich repeat region. And then the PP stands for protein phosphatase.
So are these enzymes good enzymes or bad enzymes? Do they help cancer or hurt cancer?
This would be viewed as a beneficial enzyme that kind of shuts down cancer growing pathways. Now, in normal cells, say your immune system, oftentimes you need them to proliferate and grow. And so they'll utilize these kind of pathways that allow them to turn on. But you'll need to regulate those in normal cells and turn them off when the cells recognize that it's time to turn them off. But in cancer, you'll lose something like the FLIP2 phosphatase, and those pathways will be constitutively on.
I see. Now, what has happened since with phosphatase? This was how many years ago that you were in San Diego?
I was in San Diego from 2002 to 2008 for my PhD, and then I did a postdoc at the Salk Institute with Tony Hunter for 2 years until 2010. There has been a lot of development. It was the first paper describing this enzyme. Since then, I think there's been about 200 publications on this enzyme. So it does seem to be really critical for many different physiological processes based on that, such as regulating metabolism. But in the context of cancer, it seems to be really important for cancer such as prostate cancer.
Well, that's good to know there's still work being done there by, it sounds like, a lot of your colleagues across the world.
Yes.
You started your first lab in 2010 at the Cancer Research United Kingdom Manchester Institute, part of the University of Manchester. What can you tell us about your time there?
So this was a fantastic experience and my first time living abroad. But just to start out, my postdoctoral mentor, Tony Hunter, he kind of highlighted these institutes that are in the UK that sort of have a really great foundation for a place to start a lab. And so I became very intrigued and I went over there and interviewed and was really impressed with the institute. And I had this desire to really utilize this new frontier of research that was developing based on cancer genomics. So it's the idea that we go in and we sequence a tumor cell and compare that to a normal cell, and then look at what are the genetic abnormalities that are causing the cancer, and can we exploit that knowledge to develop better therapies. And so my time at Cancer Research UK Manchester Institute was really focused in on that and trying to identify activating mutations in enzymes such as kinases, and can we develop new therapies then to treat patients that have these activating mutations.
This is Upstate's The Informed Patient podcast. I'm your host, Amber Smith. I'm talking with Dr. John Brognard. He's an assistant professor of surgery and pharmacology at Upstate. You joined the National Cancer Institute in 2016. Did you continue your work from your lab in the UK?
To some degree, we did. We were finding, though, that these mutations that would activate enzymes were occurring at a low frequency. So translating it to patients was going to be challenging. So we started to look at different types of cancers that are driven by increased expression of enzyme promoting cancer causing genes. And based on that, you can then reach a larger cohort of patients when you develop a therapy. And so we've really focused on that going forward, and then also therapeutic design. So I did switch gears for my lab, and we started to focus much more on lung squamous cell carcinoma, head and neck squamous cell carcinoma, esophageal and ovarian cancers that are driven by these copy number alterations. And we've developed several therapeutic modalities now that are quite promising in preclinical models, and we aim to take those therapies forward to do toxicology studies, prepare an investigational new drug application to the FDA, and try these in clinical trials then in the patients that desperately need new therapies, because these are patients that really don't have good therapeutic intervention options.
So you did get back into lung cancer work, it sounds like, at least as part of it.
Yes, lung cancer has really always been a focus for me. At Cancer Research UK Manchester Institute, we were really focused in on lung adenocarcinoma and these kind of somatic mutations that may cause cancer. But then as I progressed out of there, we became much more interested in lung squamous because of what's causing that cancer is quite different. And it's these increased expression of cancer drivers versus a mutation that activates it.
Well, what sorts of research will you be doing at Upstate?
So a lot of this will continue on what I did at the National Cancer Institute, with a real focus on developing new therapies to take into patients and take into clinical trials to really help these patients. And I'm really taking on new strategies for therapeutic intervention. So we develop small molecule inhibitors for activated enzymes. We've also started to develop what's referred to as a degrader or a PROTAC. And this type of molecule will actually eliminate expression of an oncogene rather than just inhibiting its activity. And then we're also developing a new class of drugs, which are referred to as antibody drug conjugates. And these basically conjugate a payload at a targeted, well, in our case, we use targeted therapies to an antibody so that you can target multiple pathways in a cancer cell to really shut those down and cause significant responses in the patients is the goal.
How close do you think we are to having a cure for cancer?
I think we're there for certain types of cancers, right? So it's interesting. There was a clinical trial that just came out. And if you have these mismatch repair mutations, the first therapy that patients were given for the first time ever was pembrolizumab, which is an immuno-oncology agent. And I think the response rates were 46 out of 47 patients. So pembrolizumab is used to basically turn on your immune system to recognize cancer cells. So cancer cells will express things on their surface to say, I'm not a cancer cell, don't kill me. But now we have these agents that bind to that, and now the immune system can automatically be turned on to recognize those cancer cells as not normal and something that needs to be eliminated from the human body. And so these advances in these types of therapies are really game changing. I hesitate to use the cure word, but these are patients that will go in remission. There'll be no detectable tumors. So it's as close as you're probably going to get. That's not to say it won't come back in 10 years, 15 years, et cetera, but it's amazing progress. And we've seen this for breast cancer as well, where I'm sure a lot of people know people that have breast cancer that are survivors. If you have HER2 or ER positive breast cancer, the response rates are pretty phenomenal. The therapies are pretty phenomenal. So it is to some degree a controllable disease now. I think we have made significant advances for certain types of cancer. And my lab's really focused on those cancers where we haven't made those inroads yet, such as ovarian cancer. And not many therapies are working well at all for ovarian cancer. And how can we come up with something much better?
What is most gratifying about your work?
I'm not there yet, I don't think. So I would say right now we do these preclinical studies. So you basically take a tumor directly from a patient and transplant it directly into a mouse, and you monitor the response of your new therapy. And what has been most gratifying is developing these new drugs and seeing this translate into these preclinical models where we're completely eliminating the tumors in mouse models. And that's very, very hard to do. But ultimately, my goal is to get those types of therapies into patients and really improve the quality of life for patients, really increase longevity, survival rates, overall survival. And that's my ultimate goal. So I haven't reached my most gratifying. Once I get a drug into clinic and it's really helping cancer patients, I can tick that box.
Is this a field that you would recommend to high schoolers who are interested in science?
Yes, I definitely would. I have two sons myself, and a concern in the back of my mind is always what's going to happen with AI and how is this going to impact future careers. But with the sciences, in particular, the biomedical sciences, pharmacology, biochemistry, I think that the human input is invaluable, and the creativity that goes into it in the development of new therapies, understanding malignancies, whether it be cancer or Alzheimer's or whatever disease you might be studying. I think that the creativity of the human brain and our ability to kind of utilize AI to some degree and apply that is a really promising career path. And I think there'll always be diseases that are going to be challenging the human body, and we're always going to need to come up with therapies to really help these patients live healthier, happier lives that's somewhat disease-free. So I would encourage high school students. It's very rewarding. You get to work with a lot of younger students, train younger students. That's one of the most rewarding things as well. And then it always keeps you sharp too, because you're working with a lot of bright people that are around you. I would highly recommend it to high school students. Yeah.
I appreciate you making time for this interview, Dr. Brognard.
Thank you. Thank you so much for having me. And I really appreciate your time as well.
My guest has been Dr. John Brognard, an assistant professor of surgery and pharmacology at Upstate who studies cancer. The Informed Patient is a podcast covering health, science, and medicine, brought to you by Upstate Medical University in Syracuse, New York, and produced by Jim Howe, with sound engineering by Bill Brokel and graphic design by Dan Cameron. Find our archive of previous episodes at upstate.edu/informed. If you enjoyed this episode, please invite a friend to listen. You can also rate and review the Informed Patient podcast on Apple Podcasts, YouTube, or wherever you tune in. This is your host, Amber Smith, thanking you for listening.